This HepG2 model provides a relevant alternative in vitro testing system to more reliably assess the potential induction of fixed DNA damage following long-term exposure to nanomaterial, with an aim to minimize the need for testing in animals. The HepG2 spheroid model possesses the ability to remain viable and functional over a 14-day period, as well as sustain a suitable level of proliferation. This supports the testing of a range of biochemical and genotoxicity endpoints, including the micronucleus assay.
Before attempting this protocol, I suggest you do a few practice runs first. Take care when cell seeding or changing media, as these require a delicate approach.
Begin by adding 100 microliters of sterile room temperature PBS to the wells of a 96 well culture plate. Invert the lid of the plate and carefully pipette 20 microliter drops of the cell suspension into the center of each well groove of the lid. Use a multi-channel pipette, adding 2 to 4 drops at a time to ensure accuracy of placement.
Only seed 4 spheroids at a time and ensure that the pipette tips are all aligned before beginning. The angle at which you hold the multi-channel will make a difference to the way the spheroids are formed.
Make sure that the drops are centered within the grooves of the wells on the lid. Then gently flip the lid on top of the plate so that the drops hang over the wells with PBS. Incubate the plate at 37 degrees Celsius and 5% carbon dioxide for 3 days prior to spheroid transfer onto agarose. After the incubation, remove the plate from the incubator and carefully lift the lid off the plate. Discard the PBS, tap the plate to remove any residual liquid, and allow the plates to air dry.